So, as he's already mentioned, so Ricardo is the CEO of MolMed, a company based here in Milan.
Yep.
And what's important, I would say, about MolMed is that I think in 2016 you got approval for gene therapy, which is, I would say, it's particularly impressive. There are not that many gene therapies approved here in Europe. So, can you tell us a bit more about the therapy, how it works?
Yeah, sure. Thank you for the question. And Zalmoxis is a gene and cell therapy because it is a cell therapy based on gene engineering. I will not enter into too many details, but it was a long journey because you know that in the San Raffaele Hospital started a relevant part of the currently available gene and cell therapy. And they started working combining two know-how. One is the clinical know-how, so the unmet clinical need for patients affected by severe hematological malignancies, not able to find a fully matching donor. Who is familiar about this transplantation technique knows that when you infuse the bone marrow of a donor to a patient needing the transplant to solve his leukemia, lymphoma, myeloma problem, you give both stem cells and the immune system, so the T lymphocytes. If the patient is not able to find a fully matching donor, or has no time to find it because the risk is to die before finding the matching donor, the immune system of the donor creates an adverse reaction against the host called graft versus host disease that could kill the patient and cancel the benefit or expected benefit of the transplantation. What MolMed was able to do, MolMed included a suicide gene within the T lymphocytes of the donor, separating the stem cell from T lymphocyte. The stem cells are transplanted in the patient. The T lymphocyte comes to our facility. We engineer these T cells, including the suicide gene. We cultivate, multiply these cells, of course purify and so on, and we reinfuse into the patients 21 days after the transplantation. What's happened that the T cells, engineered T cells, are able to protect the patient from infection and from the leukemia relapse, which is the goal of having an immune system. But if and when the graft versus host disease occurs, we can switch off these cells with the simple infusion of ganciclovir, activating the suicide gene and killing the cells. And what is amazing, because it seems that this mechanism is very clever, and it is that selectively you kill just the replicating cells, leaving the other T cells able to fight infection and to prevent relapse of leukemia. So the not replicating cells are still alive, doing their job, while the replicating cells causing the graft versus host disease are killed. And this, in our clinical trials, compared to the per match analysis versus the registry data, works in 100% of the cases. So all the patients incurring in a graft versus host disease and treated with Zalmoxis solved their problem with the infusion of ganciclovir. This is not just a nice scientific cosmetic result, because in terms of clinical result, we moved the 1-year survival rate from 34 of the control to 51 of the treated patients. So it is a strange approach, which is a life-saving adjunctive therapy with Zalmoxis. This data opened also a new approach in authorizing a drug, because it is not a drug, a therapy, because the European agency recommended and then accepted to match a combination between phase 2 data and the first phase 3 ongoing trial patients versus a per-matched analysis, so an analysis performed by the European Society of Bone Marrow Transplantation, matching 4 patients coming from the registry with 1 patient treated with Zalmoxis, and so having a real-life comparison able to provide us with a conditional market authorization 2 years ago.
Okay. And yeah, something interesting about that is that there's been a lot of talk about cell and gene therapy, how they can have, some of them, like this huge difference in the lives of patients. That some talk about like curing certain diseases, not only in cancer, but also in like rare disease. But it seems interesting to me that there's been a lot of talk about gene therapy, I think, since the 90s. And as I mentioned, there are not that many cell and gene therapies approved. You have one of the few. I think in Europe there are only 10, about 10 right now. So, well, yeah, that I guess proves my point. But basically, I just want to know a bit. I think there have been some approvals lately. So what's your view on the field? Do you think it's changing? Are we starting to get more cell and gene therapies, or is it something that is still lagging?
I would answer definitely yes. Not is changing, it's changed. It is changed. In the early 2000s, when I was working in a more traditional biopharmaceutical industry, I was not so close to the cell and gene advanced therapies. There were a lot of doubts, a lot of questions, a lot of open issues when the risk-benefit of these therapies was considered not so convenient. So the pioneers, resilient and brave enough to continue to work in this field, arrived to the target. The real turning point, as very well stated in the Alliance for Regenerative Medicine report, that was a presentation in March this year in Amsterdam, the real turning point was the second half of 2017, with the 3 approvals of cell and gene therapies in the US, because in 2016 we had Strinvalis and Zalmoxis approved in Europe, but in the second half of 2017 they quickly approved Kymriah for Novartis, Yescarta from Kite, then acquired by Gilead, and Luxturna from Spark Therapeutics. This increased a lot the focus, the appetite for both investment funds and big pharma companies, and we have seen the investment in the field of pure cell and gene therapy increasing by 78%, 17 versus 16. A number of acquisitions, the most recent from Novartis, another $8 billion, but we have seen $11.9 billion Gilead acquiring Kite, $9 billion Celgene acquiring Juno, $11.6 billion Sanofi acquiring Bioverativ. So now it is clear, and if you go to the ASH Congress, the Hematology Congress in the US, and I'm sure in June at the ASCO, everything is about the CAR-T. So the use of the immune system to treat cancer, starting from the liquid tumors and moving, hopefully, to the solid tumors, is now the big hope for the patient. Just to tell that this first CAR-T approved in refractory relapsing children and young adult patients is able to solve 80% of the leukemia in patients with no hope for their life. So definitely, yes, there is a great appetite, a lot of challenges, but I think that we are at the turnaround.
Okay. Yeah, I definitely want to talk about those challenges, because there's a lot of talk about how some of these impressive results, like getting such a high percentage of patients practically cured of their cancer. But there's also a lot of talk, I guess, first about the safety of the therapy. There have been like several deaths reported in clinical trials, and there's also, I think, a big debate regarding the pricing, because I think the first CAR-T therapies, I think Kymriah and Yescarta, are rounding the $300,000, $400,000 each, which is, I mean, it's a big problem regarding access to these therapies. So you are also working in the CAR-T space, so how can we address these issues and make CAR-T like really a effective, safe, and accessible therapy?
Yeah, this is a very good point. We have a number of challenges. Safety is the most important because all these products promise to work, and they work, but the problem is the safety. We have a second generation of CAR-T, and we are working on the third generation. For example, our solution, the CAR-T CD44V6, but not to talk about Molmed, but we included our technology of the suicide gene also within the CAR-T. So we are able to switch off these cells if some side effect will occur. This is one of the solutions. The other one is to use very selective targets in order to target the tumor and not to affect the patient. Nevertheless, there is an additional problem of readiness. We were invited a couple of weeks ago by the committee of biotechnology of the Presidenza del Consiglio of the government to participate into a very, very relevant meeting with some of the key opinion leaders, starting from Professor Mantovani, Professor Locatelli, Professor I forgot the name, but the Professor Naldini for sure, and so on. And we were invited together with Novartis, and we have a second problem of the readiness of the hospitals, because to use these therapies, they do not have only be able to manage the treatment, but they have to have a ready intensive care department to treat eventual side effect of these therapies. So the complexity is very high. We fear that not only in Italy, but in Europe, there is not a full readiness from all the aspects, regulatory, clinical, and also pricing, to come into your second point. In pricing, I would divide the issue into two areas. One is rare diseases, and the other one is oncology and oncohematology. Rare diseases, given also my long experience with Genzyme in orphan drugs, we will have probably, when we will treat the lysosomal storage disorder with these therapies, we have to compare the cost of one infusion, which is transformative, because the ADAS kid children, after the infusion, are safe and do not need any other treatment. The first child treated with the ADAS kid in the San Raffaele is now a 24-year-old guy. They are the bubble children, the children with ADAS kid, and he is doing well. So we should compare the cost, the emerging cost of one infusion with the current cost of a chronic treatment for these patients. In the oncology, what we have discussed for our product with the Italian agency, because we also obtained a cost very aligned with Kymriah, Yescarta in the US, because the cost is 149,000 euros, not per patient, but per infusion. This means that we can go from 1 to 4 infusion, and to avoid to have patient with a cost of 600,000 euros, we agreed about a flat price independently from the number of infusions. Nevertheless, it is very expensive. How do we have to work? We have to work with the authorities, with the payers, on the avoided cost. Because we are not only increasing the survival rate of the patient, but we are close to cancel the cost of the management of the acute graft-versus-host disease, of severe infection, of the chronic graft-versus-host disease. So if we want to pay these drugs, these therapies, with the standard traditional approach, we will never fund these therapies when the number of patients will increase. If we are able to calculate, and I met the general manager of the Ministry of Health preparing the new system for managing the healthcare expenditure, if we manage the cost per patient and not the cost per treatment, probably these therapies will be affordable. There is, and I don't want to take too much time on this point, but there is an additional open discussion with the authorities, which is to create something like a leasing payment. It is not needed to have all the costs paid in one installment when you infuse the patient. But as the authorities sometimes tell us, the payment by result means I pay you just if you work. If you don't work, you give me back the money. We are proposing pay me until I work. So we can dilute the payment for, I don't want to say 50 years, but 10 years. I'm quite sure that the company would be happier to have 1 million paid in 10 years than 150,000 euros in one shot. So there is a combination of open discussion that we have to find to make this sustainable. Of course, the new frontier for the CAR-T is a allogeneic CAR-T, which will cut 1-0 to the cost of manufacturing of these products.
Okay, so how much do you think the cost could be? Like if we use allogeneic therapy and even doing like some pricing schemes, like how low could the price for such a therapy be?
With the autologous, I think that we can reduce the manufacturing cost because you know, just to be clear, the quality control activity that a company, traditional pharmaceutical company, has to perform to release a batch of 40,000 boxes of a drug, we have to perform to release one batch for one patient. So the 250 pages that a traditional company has to produce for a batch of 40,000, 60,000 boxes, we have to produce for one batch, and sometimes for one patient we need 2 batches. So we have to replicate. So the cost can be reduced by automatization and some other new technologies by 15–30% from now. The real reduction of the real size reduction can be performed just if the allogeneic CAR-T therapies will work, because this is not produced for a single patient, but you can produce a batch, not creating immune reaction in a number of patients. This would significantly reduce the cost. Currently, there are some CAR-T, allogeneic CAR-T under development. One of our clients, Selectis in France, is working on this. They have the first clinical data. The only authorized CAR-T therapies at the moment are autologous. So it is a long way.
Yeah, well, at least we are starting getting there, right? And before I go on, are there any questions for Ricardo? No? Okay. Well, some other topic I also think it's very interesting to touch here is about biotech in Italy, because I mean you're an example of a company doing cell and gene therapy in Italy, and I think there are also other therapies. For example, Strimbelis is also used here at the San Raffaele Hospital. So I just want to get, like, from your experience working here, how's the state of biotechnology and particularly cell and gene therapy here in Italy, I guess, as compared to other countries in Europe?
I would say this is, the answer is it is black and white. We have 3 of the 5 cell and gene therapies approved in Europe coming from Italian research. They are Strimbelis, Dalmoxis, and Oloclar from Chiesi, the center of Modena, the center of Ferrari Research Center. We have some new startup companies coming from hospital. We have some center of excellence. The name I forgot is Professor Biondi. I apologize, with Professor Biondi of the San Gerardo in Monza, but Professor Locatelli with the Bambin Gesù. There we are able to manufacture, to produce, and to treat children with CAR-T produced internally. So we certainly have the scientific know-how. Is usually in Italy, this is true also for the more traditional biotechnology and also for the more traditional pharmacology. We are very good in science, very good in publication with a great impact factor. I think that Professor Mantovani is one of the most recognized scientists in the immunology around the world. We are a little bit less good in patent, and maybe because of the environment, maybe because of the attitude, we are not so good in industrializing, in making this good science moving to an industrial approach. We identified some key points. One is the technology transfer. So we believe that what is really needed in Italy is to make science and capital talking together with the same language. We have a map of the technology transfer of the public university research institutes, and they are small, with very few resources available, and with a more legal and administrative attitude, while they need larger organization and science and business knowledge. So if we are able to catalyze, and I think that there are a lot of initiatives in this last period, since a couple of years, this year they are increasing, to make the capital, because you know that the money have no frontiers, so we can work with Japanese money, with European, with US money. They go where there is a good science, but also a good business plan. If we are able to create an appealing ecosystem from a bureaucratic fiscal point of view and increasing our technology transferability, I believe that we have a great chance in this country, because you know that now we are in the era not of the open research, but we are in the networking era. So when I started working, unfortunately 32 years ago, the leaders of the big pharma companies were saying, what is not discovered in-house, in home, is not interesting for us. Now everybody around the world says 80% of what we will go to launch in the next 15, 20 years will be discovered outside. So my opinion, we will never be able to create Roche or Novartis, Pfizer or GSK in Italy. It is too late to have a big one. But we can leverage on this new approach to have a number of Italian companies, very good in science, cooperating with the larger companies, starting from the rare disease to the immunology, to the neurology, to the multiple sclerosis, and to the oncology and hematology, of course.
Okay. Any final questions before we finish? Yes, please.
Thank you for your presentation, Mr. Consigliere, and congratulations for what you've done at the moment. Really, we are proud as Italian that a company like yours company has made such a success. A question just raised because you mentioned patent in your talk. Now, we are using in pharma business, small molecule, biologics, we have a patent. And that's the basis for the business to grow up and make an investment. It works, it doesn't work, but at least we have this protection. My impression in gene therapy is the snappy situation about patent. What really you patent? What really you have when you have a gene technology? Because you can't patent everything, because many things that have already been discovered. You're just taking what has been discovered, it's previous art. CAR-T started in the 80s in Israel. It took 25 years to come where we see it today. And this years, many things has been already public and may not anymore patentable. So how is in gene therapy the patent issues managed?
This is a very, very good point, very well taken point. When I spoke about the patent, I was talking in general about the healthcare industry in the country. On cell and gene, you are completely right. A cell is a cell. You cannot patent a cell. The antibody is ready on the market. You can buy wherever you want, not wherever, but in many places you can have. And then you have to build. So this is a absolutely know-how intensive place where I forecast, I hope to be wrong, we will see in the future a lot of litigation. So bread for lawyers, there is some friend of mine that is very happy, but a lot of litigation, because how can I demonstrate I started before you or vice versa? So I think that, and there will be also an issue between the private companies and the GMP authorized public companies, because when I said about Professor Locatelli, Professor Biondi, so this authorized GMP in the country, they work without any patent, so they just do what they do. So I think that the real challenge will be the know-how and the size and in the scale, scalability, when the numbers will become not the very small numbers of the other kid, where we expect to have 10 to 12 children, new children in Europe per year, so negligible numbers. When we will go to the hundreds, to the thousands, I think that, and another open challenge is not only the price, the regulatory and the safety, but it's also the manufacturing capabilities, the capacity around the world. The demand in the close future will be higher than the offer. So you have seen today, Lonza announced the opening of the largest cell and gene factory facility in Texas, 23,000 square meters, 200 employees when they will conclude the population of the company. But you are very right that this is an open issue. You can patent, for example, the suicide gene, the process, but it will be a bit a nightmare in the future. You are very right.
Thank you.
Okay. Yeah, if there are no more questions, thank you. Ricardo, thanks a lot.
More than welcome. I hope that the time